Okay, good morning, good afternoon, good evening. So, kung unsa mang oras ninyo ni abrihan, a good day everyone. So, this is our first lecture video for our first unta na meeting for the created bacteriology. So, welcome to your second round of bacteriology lecture. So, hopefully we get to finish this with flying colors naman na, no? So the purpose of having this discussion is to introduce you to microbiology. So technically, hindi na i-introduce, no? I-review na lang natin ano yung mga early beginnings of microbiology as a study. Okay? So what is microbiology again? Alam kong alam niyo na ito. So taken from three terms dyan. So you can see my micro, bio, and logi. So this would mean microorganisms, living, and study. So to put them together, microbiology is the study of small living microorganisms. Okay, so why do we need to study them? Okay, so if you look at the pie chart, you'll see that a large portion of the pie chart are beneficial organisms. So napaka-importanting malaman natin ano yung benefits that we could get from microorganisms. So first, no, we have normal microbiota inside our body. Okay, so hindi sila nandun lang para makikain, di ba? So they are present in our system because they're able to help us also with the metabolic processes that we have, no? Like for example, si E. coli. E. coli is of course thriving in our large intestine, but they're also able to give back, no, in the form of vitamin K. So they provide us with vitamin A, which would be very helpful in controlling bleeding. Okay, syempre, a large portion of microbes are decomposers, no? Kagaya na nito yung mga fungi, okay? So fungi or the saprophytic fungi are able to break down dead or decaying matter, no? Para yung mga products of decomposing will be used, no, by other microorganisms in the ecosystem. And then, of course, industrial. So there are a lot of things that we can get from microbes, no? Like vaccines, for example, and medicines. When we make vaccines, we put in, no, yung actual microorganisms mismo, no, live attenuated, or their toxins, okay? And then for medicines, diba, like penicillin or streptomycin, those are antibiotics that are taken or extracted from microbes themselves. Of course, in the food industry, we make use of microbes din, di ba? Like yeast, no? Para sa bread making natin, and for the beer industry, for the liquor industry. So they make use of those things para makagawa ng mga products na yan. Siyempre pa, no? Yung mga sausawan ninyo, toyo, suka, they undergo a process of fermentation, which would be facilitated by, of course, microorganisms. Okay? Siyempre, in our field, we need to study microbiology because ang focus natin is to identify, no, the cause of the patient's ailments or diseases. So, part nun are pathogenic organisms. So, kung titignan niyo yung pie chart, konting-konti lang talaga nagka-cause ng sakit. Okay? So, it's important for us to understand how these organisms cause infection, when they cause infection, and how we can stop them. Okay? And of course, okay, because pinag-aaralan na rin natin kung yung mga microorganisms inside our body, it's important for us to understand when sila dumadami, no, kung saan sila mugara, managhan. Okay? So sometimes, pag hindi natin nabantayan, this normal microbiota can, of course, overgrow. And instead of helping us, no, wake our immune system by providing endogenous antigens, minsan nagkukos patuloy sila ng problema sa immune system natin. Okay, so it's also important to review microbiology because of that purpose. And syempre pa, in na ngayon yung genetic engineering, di ba? Oh, so nakakapagpamili na tayo ng mga genes na gusto nating ilagay kung saan-saan. Okay, so part din nun, no, yung ng genetic engineering, yung basic knowledge on microbiology. Okay? So, to review, ano yung mga naintindihan na nalaman natin about microbiology. So, syempre, nandun yung mga scientists na nag-contribute to what microbiology is today. Okay? So, ito yung mga uncles ninyo. So, si Uncle Robert Hooke, no, who introduced us to the term cell. Okay? So, dahil nag-experiment siya, no, on oak bark. And then he was able to see yung mga basic structures of life, yung cell, using just that, no? Yung microscope niya sa taas, that's made of leather, wood, of course, and a little metal, okay, na parts ng kanyang microscope. Pinagawa niya yan sa friend niya. Okay? O, nakalimutan ko yung pangalan. Okay? Mamaya tanungin ko siya kung sino yan. Anyway, so yun yung contribution ni Robert Hooke, no? So he also introduced us to cell theory together with other scientists, no? Mamaya makikilala natin yung mga kaberks niya sa cell theory. Okay? And then syempre si uncle ninyong Anton. Okay? Si Tito Anton van Leeuwenhoek, who's believed to be the father of microbiology because of his contribution of identifying the three bacterial shapes, di ba? Yung spherical, yung spiral, and of course, yung rods. Nakita niyo yun. And he called them animalcules. Okay, so yung isa ninyong uncle, si Uncle Carolus Linnaeus, introduced us to the system of naming living things, right? Binomial nomenclature. So binomial, dalawang pangalan. Genus at species. Natakpan ng mukha ko dyan naman, di ba? So, uh, pasaylo ako ninyo. Okay, so genus and species, yung dalawang part ng pangalan ng microorganism or living thing. Okay, so during, of course, the early times, nalibog pa sila kung giun sa tapag-abot diri sa kalibutan. Okay, so meron silang mga theory. Okay, yung theory of abiogenesis and theory of biogenesis. So abiogenesis is also known as spontaneous generation. Ito yung mga kakosa ng mga naniniwala sa Big Bang theory. Biglang at may buhay na agad, di ba? Biglang nagkaroon ng Earth. So, biglang nagkaroon din ng microorganism. So, sabi ng spontaneous generation, ang mga living organisms daw, nanggaling sa wala lang, non-living matter. At dahil sa vital force, nagkaroon ng life. Magic, di ba? Siyempre, mahirap paniwalaan. So merong mga mga kontrabida. Itong gumawa ng alternative hypothesis or the theory of biogenesis. Sabi nila, living organisms arise from pre-existing life forms. Ito naman yung mga kaberks ni Charles Darwin, di ba? Sa theory of evolution. So nagmula daw tayo sa unggoy. Pwede nga naman. Di ba? Mas kapani-paniwala kasi kamukha natin sila. Tsak. Okay, so nakapag-shave lang tayo ng konting balahibo kaya ayan, tao na. So sila, unggoy pa rin. Okay, so sa biogenesis, hindi sila makapaniwala na nanggaling na tayo sa magic. Nag-evolve daw tayo, depende sa environment. So nag-adapt tayo, kaya naging ganito na tayo ngayon. Straight na tayo lumakad, marunong tayo mag-design ng mga hair natin. So ganun, marunong na tayong mahiya, so may damit na tayo. Okay, so nag-evolve din daw tayo. So ganun din ang microorganism. So may marami silang mga proofs, no? Mga pros and cons na pinakita nung bawat side. So tignan natin kung ano-ano yun. Okay, ayan. So umpisahan natin sa theory of abiogenesis. So kasama nila dito naman yung napakatalinong philosopher na si Aristotle. Kung ano-anong pinag-iisip nito, no? So loko-loko, sabi, nanggaling daw sa decaying material, yung mga buhay na animal. 'Di ba? So decaying material. Tapos ang catalyst, yung spontaneous action of nature. Gihuyupan lang daw sa wind pag humanhimala yung animals. To ka. Okay? Na inituo sa'yo, syempre, gaya ni John Bautista van Helmont, alagad ni Aristotle yan. Gumawa pa ng recipe for mice. Can you believe that? Kailangan lang daw ng keg. Alam niyo yung keg? Yung pinaglalagyan ng draft beer. Ayan, yung parang kahoy na, unsa tawag na barrel. Okay, so butakan lang daw to siya trapo. Kanang hugaw. The more hugaw, the better. So trapo. Pag human, butangan mo og ka ng barley or wheat. Ayan, palay, lalagyan mong palay. Tapos, hintay mo lang ng mga around 21 days. And because of the vital force, biglang magkaroon siya ng mais. Ganun. Di ba? Katawanan siya. Pero, syempre, dahil noon, hindi pa talaga nila alam kung paano nangyari na biglang may microbes. So, ganun yung pinaggagawa nilang mga theory. Tapos hindi pa sila na kontento. Dinagdagan pa ng isang kakosa nila si John Needham. So marami na kasing nag-experiment to prove them otherwise. So itong si John Needham, nag-experiment Project IA. So sabi niya, tignan nyo, magluluto ako ng broth. So ayan, ininit niya nga, no? Ay, ito pala. Pag ganun, ayan. So, nag-init siya ng broth. Pagkatapos, pinakul niya siyempre bago niya in-store. So, during the process, bukas lahat ng kanyang container. Okay? So, ininit niya nga. Tapos, pinakul niya namang walang takip. Tapos, nung store niya, wala ring takip. O di pa paano yun? Talagang papasok at papasok doon ang microbes. O, tapos, sabi niya pa, o tingnan nyo, nagka-growth. O, alang-alang, hindi magka-growth na. So, nila nag-cook, hindi magka-microbes talaga. Okay? So, on the other hand, yung naman sa mga kakos ni Charles Darwin sa theory of biogenesis, meron din silang kani-kanilang mga experiment. Gaya nitong baliktad. Ayan. Ay, saan ba? Ayan. So, kagaya ni nasa taas, si Francesco Redi. So, si Francesco actually did the first experiment, no, to counteract or dispute a biogenesis. So, yung kanya, nag-prepare siya ng, ayan, yun nga. Ayan, ganyan. Nag-prepare siya ng tatlong container. Tapos, nilagyan niya ng meat, karne. Yung isa, nilagyan niya ng takip. Yung pangalawa, may cover pero parang net lang. Okay? And then yung isa, bukas na bukas talaga. Okay? So, hinayaan nung maburo yung meat doon. So, after a few weeks, binalikan niya yung kanyang experiment. Okay? Siyempre, yung nakatakip, walang maggots. Yung medyo may takip na net, okay? May konting maggots kasi nakikita niya, bumapatong yung langaw dun sa taas ng net. Okay? So sometimes, baka nahulog yung itlog ng fly at na-shoot dun sa baba. Hindi naman makapasok yung fly, pero magtambay sila dito sa taas. Musimhot na lang siguro sa karne. And syempre, yung container na bukas, edi labas-pasok yung langaw. Ang daming maggots. Okay? So sabi niya, tignan niyo itong nakatakip, walang maggots. Kung talagang merong magic, okay, may vital force, dapat may maggots din dyan kahit na naka-close. Pero dahil naka-close siya, hindi nakapasok yung mga living organism na manganganak doon at magkaroon ng langaw, ng maggot. Okay? Pero yung bukas na bukas, ayan, labas-pasok ang langaw, nangingitlog sila doon. Kaya nagkaroon ng maggots. Sinigundahan ito ni Lazaro Spallanzani. Okay? So si Lazaro, inulit niya yung inulit niya yung experiment na ginawa ni Needham. So sabi niya, ganito kasing ginawa ni Needham. Okay? So ayan. So sabi niya, ang ginawa ni Needham, pinainit niya, pinalamig niya, tapos in-store niya, pero bukas walang cover. Kaya naman, ayan, yung huling flask niya, may growth. Dapat ganito ang ginawa niya. So ito, pinakita niya ulit. No? So inulit niya yung experiment ni Needham. Sabi niya, dapat may takip. Iinitin mo, ipapakul mo, tapos i-store mo na may takip para walang makapasok na living organism. Kaya tignan nyo, walang growth ang aking broth. Kasi naman, hindi nakapasok yung hangin. So, true enough, lumabas din yung during that time, nanalo si Antoine Lavoisier, no, na sa Nobel Prize for identifying oxygen, no? Tapos may mga kasama din siya actually. So, na-identify nila na ang vital force sa life is oxygen, which is needed by all microorganisms to survive. So, may microorganisms na nasa air. Kasi nga may oxygen dun, so buhay na buhay sila dun. So if you allow air to enter a container, talagang mag-iiwan siya doon ng microbes. Ayun. So, nanahimik sila. Tapos dinagdagan pa talaga ni tito ninyong Louis Pasteur. Okay? So si Louis Pasteur, para matigil sila, okay, ay nag-experiment din siya ng kanya. Ayan. So gumawa siya, ayan, sa baba. Ayun. Yung kanyang volumetric flask na may napakahabang neck. Sabi niya, testingan daw niya, itong gihimo pod nila. Pero, walang, hindi i-close. Kasi nga, ang dami reklamo. Kasi yung kay Spallanzani, ihang gisarado, so di daw makasulod ang vital force. O, di rin nato siradaan. Yung si Tito Luis, ayan. So, gamit niya yung flask na may mahabang neck. Nilagyan niya ng, tawag nito, ng broth. Tapos, sa isip-isip niya, kasi kung diretso lang yung neck, yung hangin papasok, nung dire-diretso din papunta sa broth. So, bright pati siya. Iyang gibend ang neck sa iyahang flask. Okay? So, gibend niya into an S-shaped neck. Okay? Tapos, hindi niya clinose yun. So, hinit niya yung broth. He allowed it to cool there. And then he stored it na walang takip. Okay? So, bukas yun, papasok yung hangin. Pero alam niyang uh, air and the organisms that may be in air may be attracted to the walls of the neck ng flask. Okay? Di ba yung static electricity? Didikit sila yung dun sa ano, ma-attract sila dun sa walls. So makapasok man yung air, pero matrap yung mga microorganisms or solid materials na nasa air. So hindi sila aabot dun sa kanyang broth. So true enough, hanggang ngayon, eto, no, yung setup ni Tito Luis ninyo. Ayan, buhay na buhay pa at napaka-clear pa ng kanyang solution. O di ba? So nasa hangin talaga yung microbes. Kaning atikon kining mga taga abiogenesis. Okay? So, sumikat talaga si Luis Pasteur dyan. At dahil na sikat siya, nagpatuloy siya sa kanyang mga contributions, okay? Na-inspire siya. So eto, kilalang-kilala na siya. So may mga grupo ng mga wine merchants, no, na nagka-problema dahil yung wine nila aslom kayo. So they went to him and asked him, baka pwedeng pag-aralan niya kung anong nangyari sa wine nila. Sayang naman di mabenta. Okay, so he was actually able to identify that microbes present, no, in the wine has caused a fermentation of the product kaya naging maasim. Na-realize niya, no, nahuli niya na may mga organisms na gustong-gusto ng sugar. At pag may sugar, they act on it by fermentation. And in the process, they produce acids. 'Di ba? So in this case, sa wine, 'di ba usually ginagamit natin grapes. Okay, so may sugar yun, 'di ba? May fructose yun kasi fruit siya. So bacteria acted on the fruit sugar and produced acetic acid, no, in return. So yun yung nakapagpa-asim sa wine nila. Okay? Yun din yung nagpapaasim ng mga pagkain. Okay? So salamat pala kay Louis Pasteur. Kaya ngayon may suka tayo, di ba? Oh, so vinegar, yun yung acetic acid na ano na ginagamit natin ngayon. Okay? So bright po ni si Uncle because hiningian din siya ng method kung paano pwedeng ah, maiwasan yung pag-ferment ng wine. Kasi nga hindi mabenta kung maasim. And so he was able to create or develop a sterilization method for products that are really sensitive to heat. Okay? And we call that now as pasteurization. After him. It was named after him. So ang ginawa niya, pinakuratan lang gud niya og init ang product kaya hindi naman natin pwedeng initin talaga masyado yung wine or dairy products. No, kasi masisira sila. Okay? Ano yung proteins, di ba, mag-coagulate. So, ang pangit. Okay? So, ganun din naman sa wine. So, masira din yung quality ng wine kung mainit masyado o nang matagal, no? So, kadali lang, pakuratan lang yun ang material para mamatay ang microbes or mag-collapse sila. Okay? Makuratan sila mo. So, that would inhibit them from multiplying and growing in the product. Okay? So, yun yung dalawang contribution ni Tito Luis Pasteur. So, syempre, sinundan naman yun ng iba pang mga scientists, no? Like kagaya ni Agustino Bassi, he was able to identify a silkworm disease. So, dati sa panahon niya, usong-uso yung silk, di ba? Oh, so, nagkasakit ang mga silkworms, hindi makapag-produce ng silk. Nagkasakit sila dahil may fungal infection. So, ganun din naman. 10 years later, 30 years later, si Pasteur ulit, bida-bida, naka-discover ulit siya ng silkworm disease. But this time, it was caused by a protozoa. Okay? And syempre, pakasama dito yung mga kaberks ni, sino to, ni Florence Nightingale. So dito, si Ignaz Semmelweis at saka si Joseph Lister, no? So si Ignaz Semmelweis promulgated or advocated rather the use of hand washing para hindi maipasa-pasa yung puerperal fever. Ito yung sakit ng mga nanganganak na babae. Na-infect yung kanilang vaginal canal. Okay? Dahil sa maduming mga kamay, no, during delivery or childbirth. And syempre, si Joseph Lister, okay, advocated the use of phenol. I-sterilize daw yung kanilang mga surgical materials in phenol para hindi ito maging carrier ng mga infectious material at ma-infect yung mga tinatahi nilang mga wounds, right? Siyempre, kasama na rin dito si Robert Koch. Okay, so si Robert Koch was able to create steps, no, experimental steps that would prove na ang mga infections are caused by infectious agents, no? At hindi ito dahil galit ang Diyos, okay, or gigabaan ta, or dahil may demonic possession or bad spirit, kaya ka nagkaroon ng infection. So siya yun, no? Tinulungan niya tayong maintindihan yung etiology ng infection. Okay? And so he called those steps Koch's postulates. Okay? Etong si Robert Koch, marami din siyang mga experiments. No? So he was able to identify the causative agent of cholera, which is Vibrio cholerae. He was able to identify the agent of TB, which is Mycobacterium tuberculosis. During his time, usong-uso ang TB. Okay? And then, syempre, yung anthrax, which is caused by Bacillus anthracis. So, let's look at the postulates. No? Ayan, yung kanyang Koch's postulate. So, he used a dead rat, no? A lab rat na patay as a source. Okay? So, gusto niyo malaman bakit namatay yung rat. So, kinuna niya ng dugo, and then kinulture niya, and then nag-microscopy siya. So, he recorded everything. Now, when he got some colonies, again, nag-microscopy siya ulit. Okay? Tapos, nung ma-establish niya na pareho, no? Yung nakita niya under the microscope, at yung galing dun sa colony, pareho. Now, naintriga siya, tignan niya kung maipasa ba ito sa ibang organism. So, he took another animal, no, a lab rat, and then injected it with the emulsified colony sample. And then, after a few days, namatay din yung second rat. Tapos, kung ano yung ginawa niya sa first rat, he also did it here in the second rat. So, he took a sample from the dead rat, and then he cultured it and did microscopic evaluation. Tapos yung mga results niya, tinignan niya, i-compare niya dun sa kanyang previous experiment. And true enough, what he saw and what he recorded in this, from the second rat was the same as what he recorded from the first rat. No? So those are the Koch's postulates. However, there are exceptions to these postulates. Okay? Like for example, ang microorganism ay hindi makulture, non-culturable organism. So the Koch's postulates will not apply. Secondly, if for example, yung source, no, yung kanyang carrier is asymptomatic, walang manifestations, hindi naman namatay. Eh, wala din siyang signs and symptoms. And so, ang hirap, di ba? Ang hirap na i-follow through ang Koch's postulates. Another is when ang kanyang infection is caused by a variety of causative agents. No? Saksak sinagol ang nag-cause ng kanyang infection. And so, hindi rin ma-apply ang Koch's postulates. Okay? So, syempre, isa sa mga major contributions are given by Edward Jenner, si uncle ninyong Edward, na nag-contribute nung vaccination. Okay? So, dahil sa experiment niya on the cowpox virus and the smallpox virus, he was actually able to help eradicate smallpox. Okay? And he was able to establish that through vaccination, we are able to acquire a sort of protection called immunity. Okay? And then, of course, the birth of modern chemotherapy. So when we say chemotherapy, the treatment is done using different types of chemicals. So chemotherapeutic agents may be synthetic or they could be natural. Pag sinabing natural, it could be halamang gamot, yeah, no? Or it could be taken from microbes themselves. Okay, so na-process lang natin. And then pwede rin naman yung synthetic gawa sa laboratory. Okay, so one of the earliest types of chemotherapeutic agents was quinine, which was extracted from a tree bark. And it was used for the treatment, no, and prophylactic treatment for malaria. Okay? And then si Paul Ehrlich was able to develop salvarsan, which is a derivative of arsenic, and it's used for the treatment of syphilis, no, during that time. And syempre, a few years later, na-discover naman ni Alexander Fleming accidentally ang penicillin. Okay? And by 1940s, yung penicillin na na-discover niya ay na-mass produce na. And then syempre around this time also, nag-start na silang nag-synthesize ng mga synthetic drugs, no, like sulfonamides. Okay? And of course, dahil palaki ng palaki ang pamilyang microbiology, the scientists, no, deemed it important, okay, na i-separate yung focus, no, ng microbiology. So those who'd like to focus on bacteria, meron tayong bacteriology. For fungi, we have mycology. For parasites, we have parasitology. For algae, we have phycology. And of course, for viruses, we have virology. Okay? So during this time also, nagkaroon na nung identification methods for microbes, di ba? So after na-establish yung Koch's postulates, scientists started to identify microbes in different ways. One of the methods of identification that was introduced was through immunology by Rebecca Lansfield. Oh, ito si Tita Becky. Okay? Yung tita ninyong tumulong sa atin para makapag-differentiate tayo ng Streptococcus, di ba? Siya yung nag-introduce sa atin ng C-reactive protein, no, na hinahanap natin to categorize our streptococcal organism based on Lansfield's classification. Ito yun. Okay, kaya meron tayo ng Group A, Group B, di ba? Group C, G, F. Ang daming groups na ngayon ng Streptococcus. Ayan, dahil yan kay Tita Becky. Okay, and syempre Paul Berg introduced us to recombinant DNA. Dito na nag-umpisa, no, yung kanilang pagkahilig sa molecular identification. Ayan, no? So, dito na hinay-hinay na ipanganak ang PCR at slowly it has been improved to what it is today. Okay? So, bunga ng kaalaman sa recombinant DNA, nagkaroon din tayo ng developments in biotechnology. So, now we have genetic engineering, right? We have gene therapy. So, gene therapy, ito yung mga pangmayaman ito, di ba? Pampahaba ng buhay. Dinadala sa Germany, kung hindi sa China, for gene therapy. Okay? Genetic engineering, pwede ka na mamili, no? Kung gusto mo nung blonde blue-eyed, okay? Straight hair, kung ano-ano pa, matangos ang ilong, matangkad, yung mga traits na ganyan, pwede nang piliin, tapos isaksak mo dun sa, ano tawag niyan, sa genes, no? Na i-implant natin for artificial insemination. Di ba? Oh. And then yung GMOs ngayon, kagaya niyan, no? Yung kamatis. O, tamo. Yung kamatis dati, ganyan lang kaliit. Tapos ngayon, yun ang isa, di ba? Ang laki na. So, yun ang sarap ilagay sa mga burgers dahil bilog na bilog at napakalaking kamatis. Okay? And then, of course, we now are able to make use of microbes for helping us clean and treat our environment. So we call that bioremediation. So we can use Nitrosomonas and Nitrobacter, no, for oil spills. Ayan. Mga lipophilic na mga organisms to. So kinakain nila yung spilled oil. Okay? And then, of course, we have biological insecticides, gaya ng Bacillus thuringiensis. So, genes of Bacillus thuringiensis are inserted into genes of plants, okay? Gaya ng mais, no? So, nilalagyan natin siya ng genes ng Bacillus thuringiensis to inhibit yung mga parasites sa tanim, gaya ng mga caterpillar, mga uod, ganyan. So, hindi na nila makakain yung mais dahil meron siyang tawag niyan yung inborn, bali na biological insecticide naman. Okay? So moving on to bacterial structure. So review na lang natin to, no? Bibilisan natin, di na tayo take masyado ng time dahil alam na ninyo kung ano itong mga structures na to. Okay, to start off, we have the different types of bacteria. Okay, know that they are unicellular, so one cell is made up of one living organism. Okay, it's mostly free living, but no, as small as they are, they contain both DNA and RNA, and they reproduce by binary fission. Okay? Kasama, no, sa pamilyang bacteria, yung Archaebacteria. Kaya lang, ito yung mga ancient types of bacteria. They usually live in really extreme environments, no? Very salty, very init, ayan. So ito yung mga klase ng bacteria na matagal nang nag-survive dito sa Earth. Okay? At saka iba-iba yung shape nila. Meron mga square shape na bacteria, meron ng star shape na bacteria. Ayan, no? Pero yung ating concern is the eubacteria or the true bacteria, which could be, no, ayan, baliktad, ito. So, meron tayo nung spherical sa taas, we have the bacilli, and of course, itong ating spirals. Okay, so, pag sinabing morphology, kasama dito yung kanyang shape, yung size, yung arrangement. So, alam na natin may tatlong shapes. Ano naman yung size nila? So, ang size nila are usually between 0.2 to 2 micrometers in diameter and around 1 to 10 micrometers in length. The largest is Margarita namibiensis, which can measure around 100 to 750 micra. So, ang laki niya, nakikita natin through the use of the microscope. Okay? So, eto siya. They are like, ang pangalan niya is based on their morphology. So, Margarita means, Thio means sulfur. Okay? Margarita means pearl. So, sulfur-like pearls, they were found in Namibia. Okay? Off the shore of Namibia. So, Namibiensis. Ayan. So, yun yung etiology ng pangalan niya. Okay? Sa shape naman, again, nandun yung ating mga spherical, gaya niyan, pabilog, yung rods, pahaba, and of course, yung mga kulot or spirals. Okay? So, apart from size, shape, part of the morphology is the arrangement. So, sa cocci, sa spherical, they can be arranged in pairs or diplococci. Ang pinakamagandang example nito yung Neisseria, no? Diplococci. And then, syempre, in grape-like clusters, gaya nitong orange dyan, staphylococci. And then, ito, no, yung in chains, that's streptococci. Actually, yung streptococci, guys, diplococci yan sila. Tapos, nag-form lang sila ng chain. Okay? And then, meron tayo nitong tetrads, tag-aapat, or kung tag-8 sila, we call that octads or sarcinae. Okay, sa bacilli naman, meron tayo nung most of the time single, isa-isa lang. Pero naapod ng diplobacilli. Okay, sama-ani. So note, ang attachment nila is sa polar ends. Okay, hindi yung pag ganun. Okay, diliana. Yan, hindi ganyan. Rather, ganun ang kanilang attachment. Ang streptobacilli naman, in chains. Okay? And then si Coccobacilli, ito yung mga rod shape na medyo punggok. Okay, tambok, murag suriso. And then palisade arrangement, ito yung side-by-side na arrangement. Parang ganyan, no? Bacilli na magkakatabi-tabi. So, fence-like arrangement. Okay? Ang spirilla natin, guys, ito yung mga kulot. Yung spirals natin, ito yung mga kulot. Pero may dalawang klase. So, we have the spiral, the spirilla, gaya nung nasa top, no? 1 or 2 coils lang yan. And then we have the spirochetes, gaya nitong dalawa. So, pwedeng yung loose coils and pwede rin naman yung rigid coils. Okay? So, sa cell types, alam nating merong dalawang klaseng cell types, right? May prokaryotic o yung mga primitive types and may eukaryotic o yung mga kompleto na. So, pag sinabi mong eukaryotic, kompleto sila kasi meron silang mga organelles and syempre pa may nucleus. Dito sa prokaryotic cell, may genetic material naman, kaya lang walang balot. Okay? So, ang tawag sa kanila ay primitive kasi nakahubad yung kanilang genetic material. No, nucleoid lang ang tawag natin dun sa area kung asa nagtapok ang chromosomes or ang genes. So, wala kayo siya organelles, just some vacuoles and ribosomes. No? So, let's start with the first structure. So, galing sa labas, yung flagella. Okay, so, alam na ninyo na ang flagella ay organ of correct, locomotion. So the main protein here is flagellin, which is also used in serologic testing as the H antigen. Okay? So ang parts ng ating flagella, yung filament mismo, yung katawan, yung hook, no, yun yung naka-anchor sa bacterial cell hanggang nakabaon yan dun sa cell membrane niya. Okay? So we can actually identify microorganisms based on their flagella. Okay? So, doon, okay, yung pinaka-nauna, isa lang ang kanyang structure, isa lang ang kanyang flagella. So, we call that monotrichus. Ito naman, sa baba, okay, dalawa, right? Saka each polar end, that's amphitricus. Okay? Pwede namang tig-isa lang on each polar end. Pwede ring tuft on each polar end. And then, yung nandun sa kabilang side, yung green, okay, that's Lophotrichus. So, there's a tuft, okay, grupo ng flagella found on just one polar end. And then, syempre, yung nasa pinakababa, that's Peritrichus. Yung buong palibot niya na puno ng flagella. Okay? So, ang may flagella lang guys, sina bacilli at saka si spiral. Okay? Si ah Kokay, wala yang flagella. Ano sila, they're non-motile. Okay? So, kay spirals, aside sa flagella, naapod ni siya ay internal flagella or axial filaments. Pwede rin namang tawagin periplasmic filaments. So, ito yung nakapulupot sa body gaya niyan, no? So, nakapulupot yung kanyang internal flagella around the organism. Gaya nito, ayan. So, nakapulupot yung kanyang internal flagella. So, the internal flagella would contract and expand, moving the organism. So, parang spring, di ba? Ayan, parang slinky. Okay, so, pang move ng ating spiral. So, kanipang spiral lang gini siya. Wait lang. Okay, next we have the appendages for attachment. Ano yung mga ginagamit ng microorganisms natin para maka-attach, makadikit sa membranes natin, at makakolonize sa ating system? So, we call that the fimbriae. So, ang fimbriae, ito yung mga parang balahibo ng organism. So, ayan, no? Di ba? Klaro kaayo. No, pag phase contrast kuyo kaayo ang organism. So, kita natin yung kanyang mga fine, bristle-like na structure. So, pang dikit mo rin katong velcro gani. No? So, dumidikit, ayan. Gaya nito sa baba, nakakapagdikit sila sa membrane natin. Okay? Now, the other type of fimbriae, no, by the way, fimbriae is also called the common pili. Okay? Marami. The other type is called the sex pili. So, pag sex pili, isa lang na siya per organism. Maragog tin-tin. So, isa lang pagkabuo. Kaya ngilad pag kayo og daghag, tin-tin. Okay? So, isa lang. So, this one, ang sex pili is an appendage for mating. Gaya nitong ginagawa nitong tatlong to. Oh, yan, no? So, tig isa-isa, no? Na nakakapag-connect sila. Okay? The main structure, the main protein here is pilin. Okay? Now, the sex pili, again, is also seen in gram-negative organism. Huwag kalimutan. Okay? So, ang function, again, ng sex pili is for conjugation. Pero may mga times man na pwede siyang nagagamit for adhesion. Pero hindi ito kagaya ng fimbriae na pwedeng forever, no? Pangdikit dun. Ito, panandali ang adhesion lang. Okay? Then we have the glycocalyx, no? The bacterial surface coating. So, ang glycocalyx, yun yung outermost covering. It's made up mostly of sugars and some proteins. Okay? So, ang pinakamagandang function nito is that it inhibits phagocytosis. Okay? So, there are two types of glycocalyx. Nandun yung organized, okay, very tight na capsule, at nandun din naman yung very loose na slime layer. Okay? So, syempre, alam na natin si capsule ay anti-phagocytic, pero itong si slime layer, alam din natin ito yung nagkukos ng biofilm production, di ba? Kaya nababalutan nila yung mga prosthetic devices dahil sa very loosely organized yung slime layer. Okay? Then we have the cell wall. Syempre, after sa glycocalyx, pasulod, ang next covering is the cell wall. So, ang cell wall, ito yung nagbibigay ng shape ng ating organism, right? The main component here is the peptidoglycan. Okay? Ang peptidoglycan is composed of interspersed structures called ng NAM and NAG. Okay? Your N-acetylglucosamine and your N-acetylmuramic acid. So, alternating. No? Together, they are bound by peptide cross bridges. Okay? So both gram-positive and gram-negative contains this. Kaya lang, in varying concentrations or amount. Okay? The peptidoglycan actually is responsible for the staining reactions ng ating mga organisms pag nag-gram stain tayo. Okay? So, ayan ang itsura ng gram-positive cell wall. So, baga kayahang peptidoglycan. Several layers, no? 20 to 80 nanometers thick na peptidoglycan. So, kaya kahit na mag-decolorize tayo, di ba, sa gram staining, di matangtang ang crystal violet kaya tungkol sa kabaga. Kaya matanggal man yun sa top, pero yung sa baba, hindi na natin natatanggal. Dahil ilang seconds lang naman ang decolorization. Okay? Pero sa gram negative, ayan. Ayan lang ang kanyang peptidoglycan. Isang linya lang. No? Kuripot. Okay? Kaya pag nag-apply tayo ng acetone alcohol sa gram staining, matangtang dahil kaysa raman ka-layer. Although naa siya kaning iyang lipopolysaccharide layer, it's made up of lipids. Okay? Pag nalagyan niya ng alcohol, mag-disintegrate sila, di ba? Mabungkag. Mauna siya. Okay, so matanggal dahil ang crystal violet. And so, katong last stain that to which it is exposed, mauna to iyang i-absorb. Okay, kinadaot naman. Now, meron din namang mga bacterial cells na may abnormal or atypical na cell walls, gaya ni Mycobacterium and Inocardia. Although they are stained gram-positive, hindi natin masyado ito nakikita kasi yung cell wall nila merong structure na mycolic acid. Itong mycolic na acid na to is waxy substance na lisod ka ay i-penetrate sa regular stain. So, mauni po siya ang basis for acid-fast staining. No? So, depende kung makasulod ba ang stain or dili kung atong bungkag gamay ang mycolic acid. Okay? Si Mycoplasma pod, eto naman, walang cell wall. Kaya ang kanyang shape ay pleomorphic. Ayan, ang dami-dami niyang shape, iba-ibang forma dahil wala siyang cell wall. Okay? Of course, after that cell wall, nandun yung cell membrane. Importante ito, di ba? Kasi tayong humans, we only have this for the covering ng cell. Pero ang bacteria, aside sa kanilang glycocalyx, sa kanilang caps, sa kanilang cell wall, napud sila ani. Kay although wala sila yung mga organelles, right? Wala ba sila yung mitochondria, no? So they're able to produce energy through their cell membrane. Of course, responsible din ito for selective permeability. Yun naman talaga ang kanyang function. But for animals, for bacteria, this is where they breathe. This is where they get their ATP. Okay? Of course, alam na natin to ang cytoplasm, yung fluid portion ng cells natin kung saan nag-swimming ang ating mga organelles. Siyempre, chromosome, genetic material, most are single circular DNA. Pero may mga bacteria rin naman na hindi circular but linear ang kanilang genetic material. Usually kasi pag malaki, no? So gaya ng kay Borrelia burgdorferi, no? And some streptomyces, so linear ang kanilang genetic material. Okay, so again, wala silang nucleus, pero may genetic material. Gaya niyan, no? Nasa gitna lang talaga. So that portion is called the nucleoid. Okay? Ayan, gaya nito, kitang-kita mo yung nucleoid niya, yung dense part sa gitna. Okay? And of course, they have plasmids. Ang plasmids, guys, mura ni siya small portions of the genetic material. Pero, gamay man na siya. But it does have the genes that would code for antibiotic resistance, toxic metal tolerance, mga enzymes, mga toxin production, nandyan nakastore lahat. And what's worse is that these are the genetic materials na ipapasa-pasa nila sa isa't isa during conjugation. Yun. Okay? And ayan, di ba? Ribosomes natin yung ating principal factories of proteins. O, di ba? So, ang ating ribosomes, dalawa, gaya din sa bacteria, may dalawa din silang subunit. So, yung small subunit and yung kanyang large subunit. Okay? At yun yung ating, ay ganyan, yun yung ating ribosomes in action. So, yung large subunit yung green, small subunit yung yellow. So, siya yung tigplansya, di ba? Oh, ayan. So, nakita niyo yung protein na po-produce na. Ayan na. Yung protein lumalabas na. Cute, di ba? Oh. So, pagkatapos niyan, i-attach na siya sa lining para may labas yung protein. Pagka-bright. Okay? And then, of course, may inclusions and granules tayo, guys. So, these are the lunch boxes of our microbes. This may contain glycogen, this may contain gases or carboxysomes or yung polyphosphate granules. Okay? So, ito, sa taas, ito yung mga glycogen. Oh, ayan, no? Bilog-bilog. Ito naman sa Archean, Archean kay square, no? So, ayan na siya yung mga puti-puti, no? Sa sulod, those are gas vesicles that allow them to float. Okay? And syempre pa, ito yung polyphosphate or metachromatic granules. Di ba? Common yan kay saan? Naalala pa ba? So, naay ana si Coraine, na puting ana si Mycobacterium. Okay? So, we have the endospores. Ito yung parang structures that would allow the organisms to perpetuate. No? Hindi ito pang reproduce. Okay? Kagaya na sa fungi. Di ba sa fungi, ang spores is for reproduction. But for bacteria, these are for perpetuation, pangpahaba ng buhay. Okay? So, ito yung kanilang resistant structure. Now, there are two groups of organisms that are able to produce the endospores. We have the bacillus and the clostridium. Okay, mga gram-positive organisms, to gram-positive rods. Okay, now spores are formed in the process called sporulation. Okay, so ganyan yan, guys. So hintayin natin ipakita. Ayan. So, yung mother cell will replicate its genetic material. Ayan. So, nag-copy na siya. So, once na kompleto na ang pag-copy, isa-separate niya yung copy into one side, one polar side, and then lalagyan niya ng coating, which is usually made up of calcium and some acids, no? Yung kanyang calcium dipicolinate or dipicolinic acid. Okay? So once na na-form na ang spore, okay, mamatay pod ang mother cell. So usually, in cases of abnormal environments, init na kaayo, no? So dinha na matrigger ang mother cell to create, no, the spore. So once na, dinha lang na siya forever. Okay, so once na mubalik siya in a normal environment na pwede na siya mag-grow, then mag-undergo siya og germination. Ayan. No, so yan yung spore na iniluwa ng mother cell. Okay? So pag nindot ang environment, it would germinate and release the organism from inside the spore. Okay? So germination and sporulation. Okay? The spores are actually very rigid, no? Very compact. It can last as long as 250 million years. Imagine, no? So mahirap siyang sterilize. But we can do this by applying, no, pressurized steam, i-autoclave at 120 degrees Celsius for 20 to 30 minutes. Mahaba-haba compared to our usual 15 minutes autoclaving. Okay? So moving on to growth requirements, no? Bacterial growth and bacterial genetics. Ayan. So our organisms are actually classified, no? Lahat naman ng living organisms naka-classify. So there are three ways of classification. We have taxonomic, no? We have numerical taxonomy. Ito yung gamit ay computer taxonomy. We use numbers, okay? So kagaya nung sa paggamit ng VITEC. Okay, para mas madaling ma-identify. So we use numbers to identify the microbes. The basis for this is the frequency of trait sharing. How many times ba sila nag-share ng specific trait? Okay, so next we have the phylogenetic classification. Ang basis nito is genetic similarity and evolutionary relatedness. So meron tayo nung phylogenetic tree. So ayan, no, parang family tree. Okay, so ito yung mga galing sa iisang group, no? Isang kingdom. Tapos nandito yung sangay na kagaya nitong tatlo sa baba, no? So yung hedgehog, napaka-cute. Okay? So ganyan ang itsura niya. May maliit na ear, may ganong kaliit na body, may snout, may mouth, tapos may teeth na maliliit. Tapos yung next nun, so ganun ang itsura, may snout, may small ears. Okay? So nagbo-burrow din sa lupa. And then yung pinaka-last, so halos ganun din ang itsura. Magkakapamilya sila. Magkakaiba man, pero they belong to one classification, di ba? They belong to the family of rodents. Siyempre, wala yung labuting na sa taas. Okay? So, yun silang tatlo sa baba. So, phylogenetically, pareho-pareho sila ng linya ng branch. Isang ano sila, isang grupo, family of rodents. Okay? And then of course, ang pinaka-common na classification is phenotypic, no? Yung overall na relatedness ng kanilang mga structures, morphology, yung reactions nila pare-pareho. Okay? So, yan yung pinaka-common na basis natin. Now, in classification, we make use of taxa or taxon, yung mga levels of classification, hierarchical levels. So, may 7 tayo. So, we have kingdom, no? Phylum, class, order, family, genus, and species. Okay? So, alam niyo na kung anong mnemonic nito, di ba? Yung King Philip or King David came home for good spaghetti. Yan. And then, came over rather. Came over kay Oma Daddy. Came home. So, King David or King Philip came over for good spaghetti. Ganun. So, ang species, ito yung pinaka-last, right, sa taxa. Ito naman nga, yung basic unit of taxonomy. Okay, napaka-specific na nun. Okay, so it represents a specific type of organism. So, pag nag-identify tayo, we compare them with type strains or pure cultures. Usually, ang ginagamit natin sa lab is yung ATCC or American Type Culture Collection. Okay? Ang strain naman, ito yung mga subdivisions pa. Under a species, may mga subdivisions pa siya. Diba? So, it's a population of microbes descended from a species. Okay? So, may iba't ibang strain types tayo. We have yung biovars, serovars, and morphovars. Pag sinabing biovars, they differ in biochemical or physiologic differences. Gaya nung kay urea plasma urea liticum, meron siyang biovar parvo and biovar 960. So ito yung ang differences lang nun is based on how they appear biochemically or anong mga physiologic needs nila to survive, anong mga vitamins na kailangan nila. Okay? So yun lang yung difference. And then pag sinabing morphovars, ang difference nila lies sa morphology, no? Yung sa differences lang sa shape ng colony, for example, sa size ng colony, gaya ni Corynebacterium. So si Corynebacterium, meron siyang morphovar mitis, intermedius, and gravis. So ang difference ng gidani guys, kay ang kalakon sa ilahang colonies. Pag gravis, mo nitong dagko yun siya, intermedius medyo medium size. And then mitis kay mga gagmay kaayang colonies. Okay? And then si serovars naman, ang differences nila lies in their antigenic properties. Okay? So kagaya nung kay Salmonella enterica, di ba mga ilang main species? So enterica, pero na siya yung serovar typhi, serovar paratyphi, serovar cholera suis. Ang basis nun is yung differences nila sa kanilang serologic typing. Ayan, no? So again, in naming, so alam niyo na to, pag nagko-quiz tayo guys, follow the instruction. So ang i-capital lang ang first letter sa genus and the rest are small letters. Dili pa di all caps. Human, do not forget to underline. So ang scientific name sa mga libro, kung titignan niyo, naka-italics. Niya kita ka di na maka-italics ato ang handwriting no. So para sure, kay mag-underline tas ato ang scientific names. Of course, if you notice, most of the scientific names are Latinized. Hindi gawa-gawa yan. So, ni-Latinize para mukhang scientific name talaga. Okay? So, ang scientific names may be descriptive of their structure, like si Staphylococcus, di ba? Descriptive yun kasi Staphylo, alam natin, grape-like clusters. Tapos, Aureus, kasi mukhang gold ang kanyang colony, di ba? Yellow. Yun namang si Escherichia coli. So yung pangalang Escherichia, galing yun sa pangalan ng scientist na nakadiscover, si Escherich. Okay, coli would describe kung saan siya nanggaling sa colon natin. Yun, no, pangalan nila. Okay, so ang species hindi natin ina-abbreviate, hindi pwede Escherichia C. Dot. Sana? Okay, pwede lang tamag S. aureus or E. coli. Ganun. Okay? Tapos, ang species, hindi natin ginagamit yun alone, no? Mag-refer, the aureus, ano yun? Okay? The coli, ang dami kaya nila. Okay? So, hindi pwedeng gamitin alone. Pero, ang genus, pwede gamitin alone. No? Like, the Staphylococcus, yun lang sasabihin. The Streptococcus are, ganun. Or the Streptococci are, Pero hindi pwedeng, the coli are, unsa daw to? So, ang hirap. Now, identifying organisms in the laboratory makes use of a manual that would aid us in identifying unknowns, no? So, ang manual na yon ay ang Burgess Manual of Determinative Bacteriology, where tinuruan tayo how to make use of successive key features, no? Yung dichotomous key to narrow down identification. Kidaghan lagi kay sila, no? So, we make use of this one, no? Yung dichotomous key. So, nag-umpisa ka, gram reaction. Ano yung gram reaction? Positive ba? Negative ba? Okay? Tignan ko ano yung morphology. Rods ba? Or cocci ba? Or spiral? Okay? And then, try glucose fermentation. May acid production ba? Wala. Or may acid and gas? Okay? Kung wala, nag-hydrolyze ba ng urea? Kung wala, nag-produce ba ng indole? Kung hindi, nag-utilize ba ng citrate? So, kung makikita mo, the reaction can go either way. Kaya dichotomous key, dalawang reaction na pwedeng mangyari. Okay? So, ganyan tayo mag-identify ng unknown. Okay? So, moving on to physical and nutritional growth requirements ng bacteria natin. So, syempre, we try to grow them in the environment na artificial. So, we make sure na ma-provide natin lahat ng kailangan nilang nutrients to grow successfully. So, may mga major elements. Ito nga sila, no? Yung mga oxygen, carbon, nitrogen, etc. And of course, meron din namang mga trace elements like zinc, magnesium, manganese, no? Molybdenum, kailangan yun ng mga organism para mag-survive. Pero, again, hindi naman kailangan maraming-marami. Only in small amounts. Okay? So, hindi natin kailangan i-add yan kasi nandun na yan sa culture media. Okay? So, based on their physiologic requirements, pwede nating ma-identify ang mga organisms. So, those organisms that would use light as energy source, ang tawag natin doon phototrophs. Yun namang gumagamit ng chemicals, chemotrophs ang tawag natin. So, depende kung anong klase ng chemical, we can differentiate them further. Now, those that would use inorganic chemicals, we call them chemolithotrophs. Those that would use organic chemicals, we call them chemoorganotrophs. Okay? Now, based on carbon source naman, yung kailangan ng maraming carbon dioxide, we call them autotrophs or capnophiles. Okay? And those that would use organic compounds, we call them heterotrophs. Okay? Now, growth factors are also really important, okay? Pero hindi naman again masyadong madami ang kailangan. Ito lang yung mga need natin i-add kasi hindi nila kayang gawin, no? They cannot synthesize it. Like purines and pyrimidines, so you can include nucleic acids, okay? Amino acids, yung source nila ng proteins, and of course, yung vitamins, which could be their source for coenzymes and enzymes. Usually, ang nangangailangan ng mga ganito ay ang mga fastidious. Alam niyo na kung ibig sabihin yan, di ba? Yung mga OA na organisms, kailangan talaga nila ng growth factors. Okay. Ang oxygen, of course, is a very important requirement for growth. Okay? Kailangan yan lahat ng mga living organisms. Now, based on oxygen, we can also classify organisms. So, meron tayong mga obligate aerobes, yung talagang kailangan ng oxygen, no? And we also have obligate anaerobes. Katong nga dili good gusto o oxygen. Now, in between, we have organisms that are normally aerobes, pero makasurvive sila even in the presence of little or no oxygen for some time. We call them facultative anaerobes. No? So, panandaliang anaerobes. Meron din naman tayong facultative aerobes. Ito yung mga traditionally anaerobes, pero makatolerate sila o gamayng oxygen. So, we call them facultative aerobe or aerotolerant anaerobes. Of course, naapod tong mga gamay lang ilang kinahanglan. No? Need nila ang oxygen pero konti lang. We call them the microaerophils. So ito yung mga nasa gitna-gitna talaga kung tumubo. So konting-konting oxygen lang. Kasi kung nasobraan sila, they can die. Okay? Now, based on oxygen needs, meron tayo ng mga enzymes na napoproduce ng ating mga organism. So, itong sina obligate aerobes and most facultative anaerobes like the enterics can produce superoxide dismutase and catalase. So, kaya nakakapagawa tayo ng catalysis at ang nakakapagbigay lang niyan ay yung mga obligate aerobes and most of the facultative anaerobes. Wala silang peroxidase enzyme. Most aerotolerant anaerobes, no, yung kayang mabuhay kahit na may oxygen, nakaka-survive sila because they have superoxide dismutase and peroxidase. But they don't have catalase. Okay? Ang ating mga obligate anaerobes, katulad yung di mabuhay kung walay oxygen, kaning sila wala sila'y enzymes. So, they don't have all three enzymes. Okay? So, mamatay sila because they cannot break down oxygen. No? So, malumos sila because they don't have these enzymes. Okay? Then, another requirement is, of course, apart from light and yung ating oxygen, kailangan din nila ng heat, okay, para mabuhay, just like any other living organism. So, ang ating organisms na gustong-gusto nung medyo malamig, we call them psychrophiles. So, they can grow between 0 to 20 degrees Celsius. Pero meron talaga yung nakaka-survive kahit na freezing temperature. Oh, yun yung psychroduric. Okay? Mesophiles, on the other hand, can survive between 20 to 45 degrees Celsius. This is where most of your pathogenic organism would belong kasi buhay na buhay sila sa body temperature. Diba? And then meron din naman yung gusto ng medyo warm. We call them your thermophiles. No? So they can survive above 45 degrees Celsius. But naapag itong mas makasurvive yan 60 degrees Celsius, no? Yung mga hyperthermophiles natin. Kayang-kaya nila yung mainit na environment. Now, talking about requirements, syempre, water, no? Napaka-indispensable ng water. Lahat ng living organisms, kailangan yan for moisture. And of course, when we're talking about water, kasama na rin dyan yung salinity or salt. So since water is the most common solvent, salt naman is the most common solute. Okay? Now, those that would require more moisture, we call them humidophiles. Sino mang nakakapag-survive kahit na walang moisture, ang tawag sa kanila, xerophiles. Okay? So, pag sinabing salt, okay? Kasi kailangan din natin, no, para sa ating metabolic processes. So we need that, but we need it in only small or minute amounts. Pero may mga organisms na gustong-gusto nila na maalat. We call them halophiles. We have mild halophiles, we have moderate halophiles, and of course, we have extreme halophiles. Ito yung mga archaeans, no? Yung nasa Dead Sea, di ba? Alat-alat daw dun. So yun, may mga microbes din doon. They are extreme halophiles. Of course, naagoy kaning organism na hindi naman sila talaga, they would not require salt, but they could tolerate it. So ang tawag sa kanila ay halotolerant. Okay? So pag pinag-usapan yung salt and water, syempre, pag-uusapan dun yung, since sila yung solute and solvent, di ba? So pag-uusapan natin yung movement ng solute and solvent across membranes. So meron mang dalawang types of movements, remember. So yung diffusion, ito yung movement ng solute, no, across membranes, at meron naman yung osmosis, which is the movement of the solvent from one area to another. So pag diffusion, may dalawang klase. Gaya nung nasa taas, yan ay simple diffusion, no? So, common ito in small molecules like water. So, it can freely, like water, like yung mga very fine granules, okay? So, it can move from an area of greater concentration to an area of lesser concentration. Walay problema. Like coffee granules. So, for example, you have a cup of water, hot water, tapos idadrop mo yung granules. Okay? Without mixing it, mag-diffuse ragyud na sila, no? Across the, tawag nito, the cup of hot water. Pero may mga molecules na malalaki at hindi nila kaya yung simple diffusion. Kailangan nila ng assistance. At yung assistance na yun, kailangan ng merong ATP na bayad. Okay? Kaya dili mo move ang assistance kung walay energy. So here, you'll see may transporter protein. Ito yung mga ginagamit natin para sa mga carbohydrates. Kidagko na sila ng molecules, no? So, kailangan niya ng tulong para maipasok sa loob ng membrane. Okay? And for the transporter protein to move, kailangan ng ATP. Okay. So, dito naman tayo sa movement ng water, ng solvent. So, osmosis ang tawag dun sa movement ng water. So, the water moves from area of greater concentration to area of lesser concentration. So, tignan niyo yung GIF sa baba. So, yung maliliit na molecules, yun yung water molecules. So, nagmo-move sila towards the cytoplasm or which means inside the cell. So, kaning sa baba, yan yung sa environment. Yan. So, papasok sila, no? Sa membrane. Kasi konti lang yung water doon. So, all of this occur to maintain balance and homeostasis. So, pantay, para may water sa loob, papasok sila sa loob. Now, the movement of water or osmosis is actually regulated by osmotic pressure. Okay? So, kung masyado ng madami, no, tataas yung pressure para tumigil na yung movement. Yan. So, mabilis pa ang movement, pababa ang pressure dyan. So, ibig sabihin, kailangan pa ng water. Pero kung puno-puno na, stop. No? So, para magtigil, tataas yung osmotic pressure. Ayan. So, unsa nga nung mga importante ng osmosis? Importante siya syempre because it could also help us control microbes. Okay? So, remember when we're talking about osmosis or movement of molecules, we talk about the different types of solutions, right? So, you have yung solution na similar to the concentration that the cells has inside. So, isotonic, kung ano yung concentration sa labas, siya rin ang isolation, ang concentration nung nasa loob ng cell. So, walang nangyayaring abnormal. So, normal lang yung processing. In and out lang yung materials. However, kung nilagay natin yung bacterial cell sa environment na mas less ang concentration kaysa sa loob, edi abnormal yun, no? Pwedeng ikamatay ng cell. So, to maintain balance or homeostasis, so dahil mas lasaw sa gawas compared sa sulod, okay? So, musulod ang water, no? Pasulod, okay, sa cell. So, muburot ang cell. Pero remember, napaka-rigid nung cell wall. So, it can only allow a certain amount of water to go in. Pero kung walay proper na osmotic pressure, magsige siya og sulod og water. So ang ending nun, mag-burst ang cell. And that is what we call osmotic lysis or plasmoptosis. Now, what if you place the same organism in a solution na mas higher ang concentration ng solute? O di mas parat? Sa gawas, mas gamay ang tubig compared sa sulod. Okay? So remember, ang concentration ng nasa loob ng cells natin is around 0.9, 0.85 or 0.9, no? Na solvent concentration. Sa labas, kunyari, naka 1% or more, o di, parat na nakaayo. So to maintain balance, okay? Mag-contribute ang cell og water para makabawas-bawasan ng kaparat sa gawas. So ilalabas na yung water niya. So what happens to the cell this time? Okay? So dahil ilalabas na yung water niya, mag-shrink siya. Pero hindi yung buong cell. Ha? Remember, naka-cell wall yan. Okay? Yung cell wall niya, pwede nating ma-expand, pero hindi natin pwedeng ma-shrink. So, unsay mag-shrink? Ang mag-shrink kay ang iyahang cell membrane. So, the cell membrane would shrink away from the cell wall. So, pag malayo sila, yung communication nila napuputol. So, this will cause the cell to be in a bacteriostatic position. Okay? So, hindi siya makapag-multiply, hindi siya makapag-divide. Okay? Or makapag-parami. Kasi nga, malayo, hindi sila makapag-communicate properly. This process is what we call plasmolysis. Okay? So apart from ano na yung mga requirements natin, oxygen, water, carbon, 'di ba? Kailangan din naman ng water tsaka salt. So kailangan din naman ng pH, proper pH. So uh, the pH, no, gaya ng natutunan niyo before, ang kailangan lang naman ng bacteria yung slightly acidic to borderline alkaline, no? 6.5 to 7.5 or in some books, 7.2 to 7.6. Yan, no? Ang molds and yeast, acidic gid ni sila. Gusto nila ng 5 to 6. Okay? And so you can also group your organisms based on their pH requirements. Okay? Now, moving on to bacterial growth. So, alam na rin natin to, i-review na lang natin yung bacterial growth curve. Okay? So, alam natin na ang bacteria is able to reproduce by binary fission. So, mabuak lang, no? Ang mother cell, i-replicate niya lang yung kanyang genetic material. Pag nabuo na yung dalawa, ng dalawang copies ng genetic material, magka-cleave yung mother cell to form the two daughter cells. So, i-review natin yung ating bacterial growth curve. Di ba, remember, there are 4 phases of the bacterial growth curve. You have the lag phase, lag, no? Kay lag pa. Kay bago pa lang, nakapasok sa system natin. Parang jet lag lang. So, hindi pa masyadong gumagalaw-galaw yung cells natin. Wala pang division. Kasi they're still trying to synthesize their tawag nito, yung kanilang mga kailangan for growth, no? So, nagsisynthesize pa sila ng proteins, ng enzymes, ganyan. So, wala pa masyadong metabolic activity. Eventually, pag nandun na lahat ng kailangan nila, they'd start to grow exponentially. So, kung nakikita niyo, umaakyat yung ating curve. Okay? So, this is the logarithmic or the growth phase. Ayan, no? So, or exponential phase. So, dito, grabe kabilis ang kanilang pagdami. Okay? Eventually, okay, they'll come to a point where ang amount ng kanilang nutrients, okay, is pababa na at yung waste products na napoproduce nila paakyat naman. So ito yung tinatawag nating stationary phase. Here, the number of the organisms dividing is equals to the number of organisms that are dying. Eventually, aabot sa time na mahurot na gid ilang nutrients. So, inigawrot sa nutrients, magunsa sila. Mag-start na sila pangamatay, no? Mamatay na yung mga microorganisms natin. So we call this, no, pababa na yung ating curve. We call this the death or the logarithmic decline phase. So yun yung 4 phases ng ating microbial growth curve. Now, in the exponential or growth curve, doon nangyayari yung doubling time or generation time. Okay? Or the time taken for an organism to double the number. Look at that. That's E. coli. No, they'd start from 2 to 4, 1 to 2 to 4 to 8. Eventually, ayan na ang dami, ang bilis nilang dumami. No? So that's organisms in their exponential growth phase. Okay? So review. Kintahay na isa ka microorganism. Hindi mag-reproduce sila mag-undergo sila binary fission every 20 minutes. So pila daw ang mabuo in an hour. So start with 1 cell and then ang generation nila mo double sila every 20 minutes. So pila man ka 20 minutes in 1 hour, di ba? Tulo. Kay 60 minutes man. Kaya 2 to the power of 3 ang generation time niya. So ilan yon? 2 times 2 times 2. So yung yung 1, no, after 20 minutes may mong 2, ang 2 after 20 minutes may mong 4, and then for the last minute ang 4 may mong pila. Mahimo silang 8 kabuok. Okay? So mauna siya ilahang generation time. Now what happens di ay? No, what causes exponential growth to stop? Ano mag-stop to siya? Mag-stop because, sabi ko nga, ma-exhaust ang kanilang nutrients. Mahurot na. And then, ang waste products nila, magkadaghan. So, nalalason yung microorganisms natin. No? May toxin production. And of course, dahil sa toxins, pwedeng mag-change ang pH. pag nag-change ng pH, that could be deadly for your microorganisms. And so they stop growing and eventually die. So moving on to bacterial genetics. So kailangan natin ito mapag-aralan because napag-uusapan dito yung pag-transfer ng mga traits. Remember, some microbes are not really able to cause severe infections, pero because of gene transfers, conjugation, maka-acquire sila og bagong properties. Okay, so pag sinabing genetic transfer, meron tayong dalawang klase. So you have the vertical, yung mother to offspring, and binary fission usually ang process na kung saan ito nangyayari. Okay? And then we also have the horizontal gene transfer. So pag-transfer ng genes from one adult microorganism to another adult microorganism. Okay, so there are three types. Okay, so we have the conjugation or bacterial mating, transformation, and transduction. Okay? So, ayan. Tignan. So, ang F+, ito yung male version, which has the capacity to produce the sex pili. Okay? So, pag nakita niyo yung F negative, yung female, magme-mate sila. At dahil love niya, idodonate niya yung kanyang gene. So, yun yung plasmid, ha? So, ngayon, dalawa na silang naging F positive. Ayan. Nagkaroon na ngayon ng capacity yung isa na mag-produce ng sex pilus. So, that's bacterial mating. Hindi naman yan pangparami. Kung hindi, nagpapasahan lang sila ng property. So, yung F+, nag-donate ng kanyang gene that codes for production of the sex pilus. And so, yung F negative dati ay ngayon F positive na. Okay? So, this is called bacterial mating and it occurs only in enteric gram-negative bacteria. Another example of conjugation, yung pagpasa ng resistance genes. So, itong si R positive, F positive strain, may capacity to produce sex pilus and resist antibiotics. Mag-undergo sila ng conjugation at papasahan niya ngayon ng plasmid na may R positive and F positive na gene. So ngayon, yung dati wala, ngayon ay pwede nang mag-produce ng sex pilus at nakakapag-resist na ng antibiotics. So ganun yun, guys. So pwede na lang na ipasa-pasa yung mga powers nilang ganyan through conjugation. Okay? Another method of horizontal gene transfer is transformation. Okay? Transformation means you acquire, the microbe is able to acquire genetic material from the environment. Pero only those who are competent are able to perform this method of gene transfer. And how do we know competent sila? They are competent because they have the REC-A protein. So this is the enzyme that would allow the organism to absorb genetic material from the environment. Gaya niya, no? So namatay yung cell, nagkalat yung kanyang genetic material. Ito yung competent na bacteria. May REC-A protein siya. And so masasagap niya yung DNA fragment. So ayan, dahil may REC-A, pwede niya ngayong kunin yun at imimix niya dun sa genetic material niya. Ayan. So na-transform siya, nakakuha siya ngayon ng panibagong powers. Ayan, no? So again, transformation only occur in competent bacteria. And how are they competent? They have the REC-A protein that would allow them to gather the genetic material and paste it into their chromosomes or genes. The third type is transduction. Ito yung transfer ng bacterial fragments from one bacteria to another using the bacteriophage. The bacteriophage is a virus that infects bacteria. May dalawang klase tayo. Yung lytic or virulent phage or the T phages. And then, meron din naman tayo nung temperate phage or the phage lambda. Okay, ang lytic phage, sa pangalan niya na lang, virulent siya. Pinapatay niya talaga yung kanyang host bacteria. Okay? So tignan natin kung paano sila in action. Okay? So note that there are two types of transduction, generalized and specialized transduction. Si lytic or virulent phage usually facilitates generalized transduction. Si temperate phage naman is usually active in specialized transduction. Okay, so let's start with generalized transduction. Ayan, yan si lytic phage. Ayan, nakakita ng bibiktimahin. Okay, ifa-flush na yung kanyang genome. Okay, so magagawa na siya ng kanyang mga supling dyan. So in the process of assembly, no, viral assembly, ayan, so nagawa yung proteins at mag-a-assemble, masisira yung genes o yung chromosome ng host cell. So in the formation, isa sa mga virions niya, ang dala-dala ay yung bacterial phage or bacterial DNA. So pag nakakita itong phage na ito ng bibiktimahin, ang ifa-flash na ay yung bacterial DNA. Ayan. So ma-absorb yan ngayon doon sa DNA nung panibago niyang biktima. And so kani siya na bacterial cell karon, maka-acquire na siya og panibagong powers. Okay? Dahil inabunuhan siya nung virus. Okay, so that's generalized transduction. Again, ayan, viral genome, magre-replicate siya ng proteins niya. In the process, mabungkag po ang genetic material sa host cell. So, habang nag-a-assemble yung mga virions niya, na isasama dun sa isa or sa ilan yung genes ng bacterial cell. So, pag naipasa niya yan sa iba pang bacteria, so yung bacteria ngayon na yan ay makaka-acquire ng panibagong genetic trait. Ayan. So that's how bacteria can acquire new genes, new powers through the use of the bacteriophages, so transduction. So dito naman tayo sa specialized transduction. Ayan. Ayan na si virus. Dala-dala ang kanyang viral genome. So, etong naman si temperate phage. So, meron siyang ability to incorporate its genome into the viral chromosome, producing the prophage. So, yun yung wala doon kanina, di ba? So, ito may prophage. So, pag nagparami na siya ng kanyang mga supling, kasama sa genetic material, yung part ng bacterial genome. So, ngayon, combination ang genome ng kanyang mga supling. Okay, so ang dala-dala nilang genes are both viral and bacterial gene. 'Di ba? Hybrid. Oh, so pag makahanap sila ng panibago nilang victim, so yung victim na yan ay ngayon nakakuha ng dalawang klaseng chromosome, bacterial at viral genome kasi prophage yung nakuha niya. Ayan. So, ang mga organisms that would commonly undergo specialized transduction include, eto sila, si Corynebacterium diphtheriae, hindi naman talaga siya nakakapag-cause ng severe infection. However, if it undergoes specialized transduction, okay, it could acquire the ability to produce yung paninik, yung ano, no, yung membrane production dito, pseudomembrane production sa throat. Okay? Si Streptococcus pyogenes din. Dati-rati, on its own, hindi yan nakakapag-produce ng kanyang mga erythrogenic, pyrogenic toxin. Okay? Hindi yan nakakapag kain. Di ba ito yung flesh-eating bacteria? Okay? Yung sa necrotizing fasciitis. I know, naalala nyo pa yun kasi nakakadiri yung picture na yun. No? So, hindi siya talaga nakakapag-cause ng ganung klaseng infection. But if it undergoes specialized transduction, may property siya ng bacteria plus virus. Kaya nagiging mas powerful sila. Ganon din si Clostridium botulinum and Vibrio cholerae. Iron. Okay? Now, specialized transduction is also known as lysogenic conversion. So in the process, yung prophage na form, kaya hindi lang bacterial tawag nito, hindi lang bacterial properties ang nakukuha nung second tawag nito second na host, but rather pati na rin viral genome. So they could exhibit new properties. Siyempre, immune sila sa reinfection ng similar na bacteriophage, no? Yung temperate phage. Okay? Yan. So, pag sinabi nating genetic variation, kasama na dito yung mutation. Di ba? Nag-mutate nga yun. So, through transduction, through tawag nito, through conjugation, through transformation, the bacteria would undergo some sort of mutation. Kasi na-change yung kanilang original base sequence. Okay? And of course, meron din yung mobile genetic elements, yung tinatawag nating transposons or jumping genes. Okay, ito yung mga malilikot na genes. They jump from plasmid to chromosome. Eventually, tungod sa sige nilang balhin-balhin, magdikit na lang hinuon ang plasmid. So that would further enhance yung mutation. And with mutation comes the creation of new and powerful properties or traits. So, yan yung actually reason why mabilis mag-spread ang antibiotic resistance genes ng mga microbes. Okay? So, that ends our first discussion for our created bacteriology. Again, guys, alam kong serious na kayo this time, no? So, pakinggan natin yung ating video para naman ay ma-review tayo, no? Alam kong alam niyo na to, so i-review na lang natin. Kaya medyo mabilis yung ating discussion. Ipa-fast track natin ang ating mga lectures kasi this will basically be just a review of what you already know. Okay? So, thank you guys for staying with me. Okay? For bearing with me for today. Okay? So hopefully wala tayong masyadong maraming questions, no? So again, thank you for listening.